Okay, so let's start with the histology of liver. Okay, now in histology of GIT, you have noticed that four basic layers are present, right? Everyone, I know you have studied about the four basic layers, because those four basic layers we have seen in case of esophagus, then we have seen in stomach, then small intestine and large intestine. In those part we have seen the four basic layers. Okay, but from now onwards that four basic layer will not be applicable. Okay, from now onwards all the structures which we will be discussing that are present in the abdomen, okay, in that you can't directly apply the four basic layers. Okay, so today we will be discussing about a new concept. I don't know whether you have studied it or not. Okay, now that concept is whenever we are discussing about any organ, any gland, okay, at that time you should know that it is mainly made up of two parts. What are those two parts? That is parenchyma and stroma. Okay, that is parenchyma and stroma. Now what exactly is this parenchyma and stroma? That you have to understand, because that will be needing over here in order to understand the histology of liver. Okay, and let me tell you one thing in the very beginning, histology of liver is not that simple like the layers of the GIT which we have discussed. Okay, this is quite complicated. Now before that you have to understand one simple concept. If you understand that simple concept, it will become easy for you. Okay, so the first point which I have mentioned is from now onwards all the organs whatever we will be discussing, most of the, not all, okay, in that we will see that it is divisible into two parts. Okay, what are they? Stroma and parenchyma. Okay, now what exactly is this stroma and parenchyma? In case of stroma, it is the supporting. Okay, stroma means the supporting part of that particular organ or the structure. Okay, and parenchyma means the secreting element, secretory part or the producing part. Okay, now do like this. See, stroma means supporting. So S is stroma, S is supporting. So you can take this kind of shortcut in order to remember things. Next, parenchyma, secreting or the producing part. So P for parenchyma, P for producing part. Okay. Next, you have noticed that mostly whenever we are discussing about any organ, especially situated in the abdomen, okay, either it will be intraperitoneal or it will be retroperitoneal, right? You have studied about the peritoneum and you have studied about the peritoneal folds also. So you have seen that the mostly organs either it will be peritoneal, means completely covered by peritoneum, anterior posterior aspect. Retroperitoneal means only anterior aspect is covered by peritoneum. Okay. Now apart from this, there is another point. Okay, what is that? That is, if you see the organ, if I take the example of liver, if I take the example of kidney, okay. Now you know that liver is a intraperitoneal organ. Liver is a intraperitoneal organ. Some areas of the liver, these are bare areas where peritoneum is not attached. Apart from this, the liver is connected by a connective tissue capsule that is called as Glisson's capsule. Okay. Next, when you will study about the kidney, at that time you will come to know kidney is also covered by a capsule that is called as renal capsule. Okay. So this connective tissue covering is completely encircling the gland or encircling the structure. Okay. Now from that particular connective tissue capsule, it will send septa inside the substance of the gland, which will divide it into various lobes. Okay, which will divide it into various lobes. Now if you take one particular lobe and then we will study the microscopic structure. Okay, then only we can understand about the histology of that particular organ. Okay, so in this way we have to study. So first, as I said, stroma and parenchyma. Next, it is completely covered by a connective tissue capsule. That connective tissue capsule is completely surrounding the organ. What it will do? It will send septa inside it. Along with that septa, what will happen? Blood vessel, nerves will enter. Okay. So that is the part of stroma, supporting part. Okay. What it will do? It will divide the gland into small lobes. Okay. Now we'll take one lobe and then we'll study the histology. Okay, that means the structure of one lobe and the structure of other lobes, the microscopic structures are similar. Few changes will be there. If it is there, we'll discuss. In most of the case, it is same. Okay, the same pattern will be followed for liver also. Same pattern will be followed for pancreas also. And now onwards, mostly this pattern will be followed. Okay. Next, in stroma part, the supporting part, as I said, the connective tissue capsule, septa, blood vessels, lymphatics, nerves, all those are the supporting part. Okay. Along with that, another important part is also there, which is in the supporting role, that is the framework. And in most of the cases, it is made up of reticular fibers. If you remember the reticular fibers, which we have discussed in connective tissue chapter. Okay, so all those organs, okay, the parenchyma is supported by a framework of reticular fibers. So that also comes under the supporting part. Next, what about the parenchyma? The producing part or the secretory part. So if you take one particular lobe, in that you will be getting different types of tissue or different types of cells. Okay, inside the tissue, different types of cells will be there. So they will be having some particular role in secretion or in doing other function, okay, which is the most important for that particular organ. Okay, so like this we will be segregating it and then we will study. Then only it will become easy, or else if you study the book twice, thrice, you will not understand the concept. Okay, so I hope it is clear how we will study these particular organs. From now onward, this trend will be followed. Okay, unless and until we leave the abdomen. Next, in the GIT, I hope you know that there are some offshoots of the GIT. What are those offshoots of the GIT? If you start from the beginning, the list of offshoots, for example, salivary glands are there. Okay, salivary glands, till now we have not discussed, but salivary glands are also the offshoots of GIT. Next, if you come below, in the abdomen, you are getting the liver, gallbladder. These are offshoots of GIT. Then pancreas. Okay, these are the offshoots of GIT. That means they are not directly part of GIT, but they are supporting the process of digestion, the process of metabolism. Okay, the process of absorption. Okay, all those things are supported by them, but they are not directly the part of GIT. Okay, so liver is also a offshoot of the GIT. Okay, so if you see the liver, it is an accessory gland in the gastrointestinal system, which is made up of stroma and parenchyma. You see. So, as I said, liver is an accessory gland in GIT, and how it is made? It is made up of two components, that is stroma and parenchyma. Okay, so this stroma and parenchyma, what I have discussed just now. Now, if you see the outer surface of the liver, okay, it is covered by what? It is covered by peritoneum, because liver is an intraperitoneal organ. Few areas are there where peritoneum are absent, and those are called as bare area of the liver. Already you have seen the bare area of the liver. Next, beneath the serous coat, okay, a connective tissue covering is present. And what is the name of that covering? It is called as Glisson's capsule. Just below the serous coat, which is covering the liver, a connective tissue capsule is present. What is the name of that capsule? Glisson's capsule. Okay. Now, from this Glisson's capsule, okay, as I said, branching septa or trabeculae, that will go inside the gland, or that will go inside the liver, and along with that, blood vessels, nerves, lymphatics, they are also passing. And as I said, that will divide the liver into numerous lobes. Okay. So, along with the branching septa, radicals of the portal vein, hepatic artery, bile ductules run together within this septa, okay, which is covered by Glisson's capsule to form the portal triad. So this is very important structure which we have to see under microscope, that is the portal triad. Triad means three things. So how it is formed? As you can see, it is formed by the radicals of the portal vein, hepatic artery and bile ductules. Okay, so together along with the covering of the Glisson capsule, as I said, they will enter inside the liver. Okay, now this structure together they are called as portal triad. Okay, now in case of liver, as I said, two parts are there. What are these? Stroma and parenchyma. Now if you see the stroma, as I said, first of all the Glisson's capsule will be there. Blood vessels, nerve, lymphatics, bile ducts travel within the connective tissue stroma. Okay, now it will extensively branch and divide the parenchyma. So this particular Glisson's capsule, what it will do, it will extensively divide the parenchyma into the basis of structural unit called the classical liver lobule. So just now I mentioned this a particular branching septa which is coming from the capsule, connective tissue capsule. What it will do? It will divide the liver into various lobes. First we will see about the lobes, then we will go to the lobules. Okay. Next, for another part is there which is supporting the parenchyma, that is the reticular fibers. Now reticular fibers already we have studied, these are nothing but the type 3 collagen fibers. Okay. Now here these reticular fibers form a network that is called reticulum to support the liver parenchyma. Okay. So along with this connective tissue septa, blood vessels, nerves, lymphatics, this reticular fibers are also there which is supporting the parenchyma. Okay. Next, if you see the parenchyma, what do you think? As just now I mentioned, in case of parenchyma, specialized cells are there or a specialized tissue will be there, okay, which will do some important function. Now, here in this case, in case of liver, which cells will be there? Hepatocytes are present. Okay, and along with the hepatocyte, other few types of cells are also there, but majority, if you see, it is made up of hepatocytes. Okay, so here parenchyma, it consists of hepatocytes. Now, how the hepatocytes are arranged? If you see the arrangement of the hepatocytes, these are in the form of plates or cords. Okay, and the hepatocytes are forming a layer. Okay, the way it is arranged inside the liver, the hepatocyte, if you see, they are in the form of layer. That's why it is called as laminae. Okay, they are in the form of a layer, and this layer where the hepatocytes are present, okay, it is one cell thing. That means only one layer of cell, they are arranged in the form of cords and plates, which is forming a layer or also called as laminae. Okay. Now, if you see the layers, suppose one layer of hepatocytes are there, another layer of hepatocytes are there. In between these two layers, a small space is there. Okay, and that space is called as hepatic sinusoids. The space which is present, that is called as hepatic sinusoids. Okay, so this is the orientation of the stroma and parenchyma of the liver. Okay. So hepatic sinusoids, as I have mentioned, present between the hepatic laminae and also between the cords of the hepatocytes. Okay, so hepatic laminae means the layers of the hepatocyte cells which are present. In between that, the sinusoid which is present, it is called as hepatic sinusoid. Okay. Now there is a peculiarity of the liver is that in this particular sinusoids, both arterial blood and venous blood, they are mixing. Okay. I don't know whether you have studied this or not. In case of liver, the hepatic sinusoid, arterial blood and venous blood, they are mixing. This is the only place in our body where arterial blood and venous blood is mixing. Okay, you will not see this kind of phenomena anywhere in our body. Okay. Now, adjacent sinusoids, what they will do? They will communicate with each other to maintain the intrasinusoidal pressure. Okay. Now, blood from the sinusoids open into the central vein that occupies the center of the lobule. Okay. And in case of hepatic sinusoids, if you see the blood flow, it is centripetal direction. Okay. Now, what is the meaning of this centripetal direction? See, sinusoid means blood will flow through the sinusoid. Okay. Now, in case of liver, it is centripetal in direction. That means from periphery, the blood is going towards the center. That is the centripetal direction. Now, finally, the central vein will drain into the hepatic vein. Okay. This is the way how the blood is coming or leaving the liver. Now let me show you the diagram. Now in here you can see the septum. First of all, just see the septum. Okay, as I said, the septum which is going from the capsule and dividing the substance or dividing the organ into lobes. Okay, so here you can see this is the septum. Next, what are these? These are nothing but the hepatocytes. Now, how the hepatocytes are arranged? These are in the form of cords. Okay, branching cords, and this appears in the form of laminae or in the form of layer. So see here one layer of cells, here one layer of cell. Now in between this, the space which is there, this is called as sinusoids. Okay. See, these are this is sinusoid, this is sinusoid, this is sinusoid. Okay. Now, along with the hepatocytes, some other cells are also there. As you can see, Kupffer cells are there. Okay, having important function, phagocytic functions. Okay. Next, those cells are also lined by endothelium. Okay, the sinusoids are, as we will go further, I will show you, those sinusoids are also lined by endothelium. Okay, so this is the orientation of the sinusoids and hepatic laminae in the liver. Okay, next the liver lobules, that means the substance of the liver is divisible into large lobes. As I said that connective tissue septa which is coming from the Glisson's capsule, what it will do? It will divide it into numerous lobes. Now each of the lobe is consisting of numerous lobules. Okay, because the septa which is entering, okay, the septa which is entering the liver, further it will branch, it will divide. Okay, though it is branching and dividing, that's why what it will do. Now suppose imagine one septa coming like this, another septa coming like this. Okay, दोनों septa के बीच में, in between these two septa, the area which is there, that is called the lobe. Okay, next this septa which is coming, this is also divide. So as it is dividing, what it will do? It will make the lobe into smaller lobules. Okay, so what we have to do? We have to pick one lobe. Okay, we'll pick one lobe and then we'll see what is present inside it. Okay, like this only we have to study about the microscopic structure of liver. Okay, so suppose you are picking one lobe, inside that 3 lobules are there. Okay, so the parenchyma of the liver is divided into 3 ways, that is 3 liver lobules are there. Now what are those 3 liver lobules? As you can see, classical liver lobule is there, portal lobule is there, and liver acinus. Okay. Now in case of classical liver lobule, if you see each and every lobule, the shape is different. Okay. Now here in case of classical lobule, it is hexagonal in shape. In case of portal lobule, you can see it is triangular in shape, and in case of liver acinus, it is elliptical in shape. Okay. So the shapes are different. Next, when we will go inside this particular lobule, okay, when we will go inside this particular lobule, okay, things will get changed. For example, if you take the classical liver lobule, in the center, here we are getting one central vein, but here we are getting one portal triad. Okay, just now I have mentioned what is this portal triad. Next, at the periphery, okay, you can see connective tissue is there, then structures of the portal triad, portal canal. Now here at the periphery, central veins are there at the corner of the triangle. Okay, so like this, as I said, if you go inside each and every lobule, the structure is to get changed. Next, in case of acinus, okay, 2 axes are there, short axis, long axis, and along with that, different zones are there. Okay, so first we will be discussing about each and every lobule. So first I will start with the classical lobule. So what is the shape of the classical lobule? Hexagonal in shape. I hope everyone you know what is hexagonal shape. Okay, so first let me show you the hexagonal shape and then we'll discuss about the classical liver lobule. So which shape is this? Hexagonal shape, right? So this is the example of classical liver lobule. Okay. So in case of classical liver lobule, if you see what is present at the center, a central vein is present. Okay. So first of all, you can see a central vein is present over here. Then from the periphery towards center, you can see the plates or cords of hepatocytes are present. Okay. In between them, the area which is there, these are called as sinusoids. These are called as sinusoids. Okay, that means the blood is flowing like this from the periphery, it will go towards the center. Okay, this is how the blood is going. So just now I mentioned in case of liver, in which direction blood is flowing, from periphery to the centripetal direction. Okay, towards the center. Next, at each and every angle, can you see the angle? Each and every angle, in case of this classical liver lobule, what is present? Portal triad is present. Portal triad is present. As you can see, the portal triad. You can see the portal vein, you can see the hepatic ductule, and you can see the hepatic artery. So in each and every angle, okay, of the hexagon, what is present? Portal triad. Now, along with the portal triad, the connective tissue covering will also be there. Okay, and this portal triad, okay, is present inside the portal canal. Okay, so portal canal is there through which these structures are passing. Okay, so this is the liver, classical liver lobule. Okay, so in case of classical liver lobule, just now I have shown you it is hexagonal in shape of mass, where at the center the central vein is present. Okay, surrounded by radiating anastomosing plates of pods of hepatocytes. Now, adjacent plates of the hepatocytes are separated by sinusoids. Okay, so I have shown you the spaces, empty spaces, whatever it was present over here, that was sinusoids, hepatic sinusoids. Now, between the adjacent classical lobule, a small quantity of connective tissue is present. Okay, within that the portal triad is present. So, in each and every angle, we have seen portal triad is present, and along with that, the portal triad is covered by this connective tissue, which is coming from the Glisson's capsule. Okay. Next, a important space is there, which very important. Okay, important space is there, which is related to the classical liver lobule, and that is called as space of Moll. Okay, that is called as space of Moll. Why this particular space is important? Because from this particular space only the lymphatics of the liver arises. Okay, the lymphatic of the liver, which is draining the liver, it arises from this space of Moll. Okay, so as you can see, the space of Moll is a space between the connective tissue stroma of the portal canal and the outermost hepatocytes. Okay, so the connective tissue stroma of the portal canal and the outermost hepatocytes, in between that the space which is there, that is called as space of Moll, and it is also called as periportal space. Now it is the site of the formation of lymph of the liver. That means lymphatics of the liver is forming from this particular space. Okay. Now in case of human being, if you see the lobules, the connective tissue is not thick. Okay, but if you see some other animals, okay, especially in case of pig, the lobules are clearly seen. Why? Because of the thick connective tissue. Okay. Now let me show you a diagram. I hope you can see the diagram. It is a very beautiful diagram where you can see the cut section, okay, of the portal triad. Okay, so you can see the structures of the portal triad. So what is this? This is the hepatic artery. Here you can see this is the portal vein, and this is the bile duct tube. Okay, so these three will form the portal triad. Okay, now just concentrate on this. Around the portal triad, can you see a layer of hepatocytes? Beautiful. You can see a layer of hepatocytes. Okay, and then you can see a layer of connective tissue also. In between these two, a small space which is present over here. Now this is called as space of Moll. Okay. Now in this case, what happened? The lymphatics of the liver is beginning from this particular space. The lymphatics of the liver. Okay. Now here you can see the direction of the flow of the blood. So already I have mentioned. Okay, in case of liver, what is the direction of the flow of the blood? Towards the center. So if you see the portal triad, where they are situated? At the periphery. From the periphery, it is going towards the central vein, which is present at the center. And this central vein, ultimately it will drain into the hepatic vein. Okay, just see the direction of the flow of the blood. Okay, now this is the sinusoid. I was talking about this. This is the sinusoid. Okay, so these are the hepatocytes. These are the hepatocytes. This is the sinusoid. Okay, now in the sinusoid you can see these are the endothelial cell, liver endothelial cell, okay, which is lining the sinusoid, and inside the sinusoid you can see the mixing of the blood is taking place. Blood which is coming from hepatic artery, blood which is coming from portal vein, here the mixing of the blood is taking place. Okay. Now around the sinusoids, along with the hepatocytes, you can see the stellate cells are also present. Okay, you can see the stellate cells are also present. Okay. Another important point, please note it down. In case of liver, the flow of blood and the flow of bile is opposite to each other. Okay. In case of liver, the flow of blood and the flow of bile is opposite to each other. What is the meaning of this? So, what do you think? How the bile will flow? Here you can see the blood from here, it is going towards the central vein. How? What about the bile? Bile will go like this. Okay, see, this is the bile duct. Okay, so after production, the bile will go like this. Okay. Now this particular part, okay, where I have shown the bile is going in opposite direction, this is the part of intrahepatic biliary system. Intrahepatic biliary system. Previously we have discussed about the EHBS, if you remember, extrahepatic biliary system. Okay. Now here this is the intrahepatic, that means inside the liver, how the bile is flowing. Okay. Now this is very important. See, I am focusing on these points because from there only MCQs are asked. Okay. So please note it down. Okay, so we have seen the classical liver lobule, then I have shown you the space of ball, and I have shown you the flow, how the blood is flowing, how the bile is flowing inside the liver. Okay, next is the, after classical liver lobule, what was next? That is the portal lobule, followed by the portal canal. Now, if you see the shape of the portal lobule, what is the shape? It is triangular. Previously, what was the shape of the classical liver lobule? Hexagonal in shape. Okay. Now, what is the shape of the portal lobule? It is triangular in shape. Okay. Now, so this is the triangular area of the liver parenchyma around each portal triad. Okay. So that means you will be getting portal triad. In between the portal triad, a triangular area is present. Now, this is also considered as the functional lobule of the liver. Okay, as the bile duct lies in the center. So here, what you will get? You will get the bile duct. Okay, or one bile ductule at the center. Okay, and central vein at the corner of the portal lobule. So triangle shape is there. So at each and every corner, what you are getting? You are getting the central vein, and at the center, what you are getting? A bile duct. Okay, this is how the portal lobule is formed, and it is also called as the functional lobule of the liver. Okay, and it is also called as the nutritional lobule of the liver because it is obtained by joining the central vein of three adjacent hepatic lobules. Okay, let me show you the diagram. So here you can see the diagram. In this diagram you can see, so what is this? What is this? This is the classical liver lobule which we have discussed just now. Okay. Now this particular, if you take three classical liver lobule, okay, in between that we are getting the portal lobule. The portal lobule is also called as nutritional lobule of the liver or functional lobule of the liver. How it is formed? First of all, it is a triangular shaped area. Okay, as you can see, it is triangular shaped area. So you can see the branch of a portal vein at the center. Okay, in the portal lobule. Okay, and see, these are three central veins which are present at the periphery in case of the portal lobule. Okay, so this is how the shape of the portal lobule and the structures of the portal lobules are present. Next, along with the portal lobule, the portal canal is also there. So through the portal canal, what is passing? The structures of the portal triad. Okay, the structures of the portal. I have shown you the portal triad, the structures of the portal triad. Now, how it will pass? It is to pass through the portal canal. So at the portal canal at the angular interval at the periphery of each lobule. Okay, if you go to the periphery of each classical liver lobule, what you will get? You will be getting the portal canal. Now this portal canal, first of all, it is filled with connective tissue, okay, which will be supporting the portal triad, the structures. Now the portal triad, it contains radical of the portal vein, hepatic arterial and interlobular bile duct. Okay, these are the structures which are present inside the portal canal. Okay, radical of the portal vein, not directly the portal vein. Okay, then hepatic arterial and interlobular bile duct. Okay. Now this structure collectively they will form the portal triad. Okay. Now blood from the branch of the portal vein and from the branch of the hepatic artery will enter the sinusoid where the mixing of the blood is taking place. Okay. And then it will pass towards the center where the central vein is present, and from the central vein ultimately it will go to the hepatic veins. Okay. Now here you can see the diagram. It is a 3D diagram of the portal canal. Okay. To see, I hope you can see this is a hexagonal shaped classical liver lobule. Okay. Previously the diagram which I have shown you, that was not 3D diagram. Now this is a 3D diagram. Okay. Now in this 3D diagram you can see the hexagonal shaped classical liver lobule. Okay, and you can see this as the portal canal. This is the portal canal. This is the portal canal. Now, through this portal canal only the structures of the portal triad are passing. Okay. Next, the last one is the portal acinus. Now, what is the shape of the portal acinus? It is diamond shaped. Okay, smaller unit of the liver tissue. So, this portal acinus is small as compared to the other lobule which we have discussed. Okay, and this is particular supplied by one hepatic artery running along the line of the junction of two hepatic lobules. Okay. So in between the two hepatic lobule, one branch of the hepatic arterial is there which will go and supply this particular diamond shaped area which is called as the portal acinus. Now two central veins lie at the end of each acinus. Okay, and this is also called as the functional unit of the liver. This portal acinus also called as the functional unit of the liver, and it is having two axes. One long axis is there and one short axis is there. Okay. So how the long axis is formed? It is the long axis of the acinus is formed by joining two central vein. Okay. So two central vein if we join, then we will be getting the long axis, and short axis is obtained by joining the two portal triads or adjacent portal triad. If you join, then you will be getting the short axis. Okay. Next, there are three zones. Okay, three zones are present of the liver acinus. Okay, because as I said, it is as I mentioned, it is supplied by branch of one hepatic arterial. Okay, so three zones are there: zone 1, 2, and 3. Now, in case of zone 1, it is near the portal triad. Zone 2, it lies in between 1 and 3, and if you see the 3, it lies near to the central vein. Okay, now these are having important clinical significance. Now here you can see the portal acinus. Okay, so see, this is the diamond shaped area. So this is supplied by one branch of the hepatic arterial. Okay, now in this particular diagram you can see this is the long axis. Okay, you can see the long axis of the portal acinus, and here you can see this is the short axis of the portal acinus. Next, the branch of the arterial which is supplying this particular area. Okay, depending on that, three zones are there: zone 1, 2, and 3. Okay, so you can see zone 1, the periportal zone, more exposed to digestive nutrients, toxin, okay, which is received from the gut, okay, and having high oxygen supply. This is the zone 1. Next, if you go to the zone 2, it is intermediate, and in case of zone 3, it is the central zone, has poor oxygen supply. So I am talking about this central zone but poor oxygen supply. Okay, and it is prone to hypoxic injuries. Okay, so this is the clinical significance of these three zones of the portal acinus. Okay, so three important diagrams in one part. As you can see, this is the classical liver lobule. Then what is this? This is the portal lobule. Okay, triangular shaped. This is the hexagonal shaped, and here you can see the diamond shaped area, that is also called as the portal acinus. Okay, now there is a important space. Okay, another important space, that is called as space of Dis. Okay, now what is the space of Dis? So in case of liver, as I have mentioned, the cells which are there, especially the hepatocytes, okay, now they are present around the hepatic sinusoids. Now if you see the sinusoids, these hepatic sinusoids, these are lined by discontinuous endothelium and discontinuous basal lamina, and along with that stellate macrophage cells are also there. Stellate cells or macrophage cells are also there. These are called as Kappa cells. Okay. So these are present along with the hepatic sinusoids. Okay. Now another important space is there, which is called as perisinusoidal space, that or also called as space of Dis. Now this sinusoidal epithelium separated from the underlying hepatocytes by a extracellular space, and that is called as space of Dis. Okay. Now what is present in the space of Dis? The hepatocytes, they are also having microvilli. Now those microvilli are projecting into the space of Dis. Okay. So the microvilli which are present of the hepatocytes, they are projecting, okay, into the space of Dis. This is the important significance of the space of Dis. Okay, but in case of liver, you will not get any significant blood-liver barrier. Okay, why? Because large gaps are present in between the endothelium, okay, and discontinuous basal lamina of the endothelium. That's why a blood-liver barrier is not present. Now, in this, I hope you can see the space of Dis, okay, or also called as perisinusoidal space here. This is the perisinusoidal space. So from here, the hepatocytes, the microvilli will be projecting over here into the space of Dis. Next, another important type of cells are also there, that is called as hepatic stellate cells or Ito cells. Okay. Now these are mesenchymal origin, and what is the main function? The main function is they are involved in the storage of vitamin A and lipids in case of liver. Okay, so till now whatever we have discussed, that was about the 3 liver lobules. Okay, next part is the intrahepatic biliary tree, okay, or intrahepatic part of the biliary tree through which the bile is flowing. Okay, so what do you think, how the bile are produced? Bile are produced by the hepatocytes. The bile which is coming into the second part of the duodenum, how they are formed? They are formed by the hepatocytes. Okay, so bile is formed from the hepatocyte. Okay, ultimately where they are reaching the bile? Once it is formed from the hepatocyte, where it is reaching? We have studied the extrahepatic biliary system into the second part of the duodenum through major duodenal papilla along with the pancreatic duct. After that only it can emulsify the fat. Okay, so that is the destination. What is the origin point? That is the hepatocytes. From hepatocytes bile production hota hai. After that ultimately where it is reaching? It is reaching the second part of the duodenum. So that is the journey of the bile from the hepatocytes to the second part of the duodenum. Okay, now some part of the bile which is traveling, that is inside the liver, and some part outside the liver. When it is outside the liver, at that time it is called as extrahepatic biliary system. Extrahepatic biliary system, that already we have discussed. Okay, now inside the liver, when it is passing, okay, at that time that is called as intrahepatic biliary system, or also called as intrahepatic biliary tree. Okay, so how much will be the intrahepatic biliary tree? Any idea? From liver, when we have discussed about the extrahepatic biliary system, I hope you remember I have started with the hepatic ducts. 2 hepatic ducts are there, right and left hepatic ducts. Okay, coming out from each lobe of the liver. Okay, they will join together to form the common hepatic duct. So those 2 hepatic ducts which are coming out from the 2 lobes of the liver, okay, from that part will be the extrahepatic biliary system. Beyond that, it is intrahepatic biliary system or intrahepatic biliary tree. See, you have to understand the concept कि कहाँ से कहाँ तक intrahepatic है और इसके बाद कहाँ से कहाँ तक extrahepatic है. Okay, so extra we have discussed inside the classroom. So today we will be focusing on the intrahepatic part. Okay, so intrahepatic part, what is the main function? That is the exocrine part of the liver. Okay. So we know that liver is secreting bile, which is done by the hepatocytes, okay, and it conveys it into the extrahepatic biliary tree with the help of right and left hepatic ducts, okay, and ultimately it reaches the second part of the duodenum. Now, in case of the intrahepatic biliary tree. What is the first part? That means from the hepatocyte, when the bile is secreted, where it will go? It will go to the bile canaliculi or bile canaliculus. Okay. So first is the bile canaliculus. So this canaliculi are the spaces present between the plasma membrane of the adjacent liver cell. Okay. So in between the plasma membrane of the liver cell, the blind space which is present, it is nothing but the bile canaliculus. Okay. So what the liver does, the liver will secrete and it will pour the bile directly into the bile canaliculus. So जो hepatocytes से जो bile निकल के आता है, directly it will be poured into the bile canaliculus. Okay. Now after bile canaliculus, okay, what we will be getting? We will be getting the intralobular ductules, or also called as canal of Herring. Intralobular ductule. Now bile canaliculi will drain the bile into the intralobular ductule. Okay. Now these are mainly lined by simple cuboidal epithelial cell. Okay, these are mainly lined by simple cuboidal epithelial cell. Okay. After that, next we are getting the interlobular duct. Intralobular, then interlobular duct. So you can see the interlobular duct. Now intralobular duct drain into the interlobular duct. Okay, run through the intralobular connective tissue septa of the portal triad. Means after this, okay, so intralobular duct will drain into the interlobular duct. Now this will go into the portal triad. Okay, where the portal canal is present. Now this, if you see these particular ducts, they are lined by simple cuboidal, and larger ducts are lined by simple columnar epithelium. Okay. Now here you can see the intrahepatic biliary tree. Okay. So bile canaliculi. Okay. After bile canaliculi, we are getting the canal of Herring. Okay. You can see canal of Herring. Then what we are getting? Interlobular ducts. Then septal ducts are there, area ducts are there, segmental ducts are there. As you can see, the diameter is increasing as we are going downwards. Okay. Then these segmental ducts will join to form the right and left hepatic ducts. Okay. So this is the right and left hepatic duct, and beyond that, it both will join to form the common hepatic duct. Okay. So this is the intrahepatic. This up to this part is the intrahepatic biliary tree. Okay, how the bile is flowing. So here what you have to notice, from where it is starting, okay, and gradually how it is draining. That means it has to come to the portal triad. Okay. In between that, the cells which are present in the duct, what is the lining? So in case of smaller duct, it is lined by the simple cuboidal epithelium. As you go further in the larger duct, it is lined by simple columnar epithelium. Okay. Now this interlobular duct, okay, interlobular duct, they will join together to form the right and left hepatic duct. Okay. Now this right and left hepatic duct will come out from the right and left lobe of the liver. Okay, and after that they will join together to form the common hepatic duct. Now that common hepatic duct is outside the liver, not inside the liver. So from common hepatic duct is the part of extrahepatic biliary system. Okay, next there are some accessory duct that is called as duct of Luschka. Okay, and in case of extrahepatic duct, already we have discussed previously. Okay, so now I am not going to mention about the extrahepatic ducts. Now this is the flow chart how the bile is flowing. So as you can see from the hepatocytes, first bile is produced, then bile will be poured directly into the bile canaliculi. From bile canaliculi, we are getting the intrahepatic ductules, or also called as canal of Herring. Okay, from there it is going to the interlobular, then intralobular bile ductules, okay, of the portal triad. After that, right and left hepatic duct, which will join together to form the common hepatic duct, and like this it is leaving the liver. So this is the flow chart how the bile is flowing. Okay. Now in the various diagrams I have shown you hepatocytes. Various times I have shown you the hepatocytes. Okay. Now exactly what is the shape of the hepatocyte? Okay. What is present inside the hepatocyte? Okay. So if you see the liver parenchyma, as I said, mainly it is made up of liver mesenchyma, it is maybe made up of liver cells, and these are called as hepatocytes. Okay. Now what are the shape? These are large polygonal shape. If you see the size 20 to 30 micrometer, okay, quite larger. Now if you see the nuclei, it is large spherical nuclei and present at the center of the cell, the nucleus. Okay, the nucleus show huge euchromatin. Okay, why? Because these cells are metabolically active, because they are synthesizing continuously. The nucleus shows 1 to 2 nucleoli. Many hepatocytes are binucleated. Lifespan is approximately 5 months, and it is having high regeneration capability. Okay, that means if some part of the liver is damaged, it is having the capacity to regenerate again. Okay, and cytoplasm is slightly acidophilic. Next, there are three surfaces of the hepatocytes. Okay, what are those three surfaces? Sinusoidal surface are there, canalicular surface are there, and intercellular surfaces are there. Three surfaces, very important. Okay. Now, if you see the sinusoidal surface, so what is the meaning of sinusoidal surface? It is facing the sinusoid. So just now we have mentioned, we have seen the hepatic sinusoid. Okay. So the part of the cell which is facing towards the hepatic sinusoid, that is called as sinusoidal surface. Okay. Now this surface of the hepatocyte shows microvilli, and which is projecting into the space of the disc. Okay. So in that particular area only the space of disc is present, and in that particular space of disc the microvilli which are coming from the sinusoidal surface, okay, they are projecting. Now the microvilli, why they are present? They increase the surface area of the cell for transport of nutrients across the hepatocytes. Okay, that's why these microvilli are present. Next, canalicular surface are there. Canalicular surface means towards the bile canaliculi or bile canaliculus. Okay. Now that forms the wall of the bile canaliculus, approximately 0.5 micrometer in diameter, along with the adjacent hepatocyte. Okay. Now the bile canaliculi are separated from remaining intercellular spaces by tight junction, and intercellular surface, that is adjacent hepatocyte, means one and another hepatocyte which are facing towards each other, it is called as intercellular surface. Okay. So like this, in case of hepatocytes, three surfaces are there. Now this is the diagram. In this diagram you can see the surfaces of the hepatocytes. Okay. Can you see the sinusoidal surface? See, this is the sinusoidal surface. See the microvilli, how they are projecting into the space of disc. Okay. Next, this is the canalicular surface. Okay. So this particular surface is also forming the wall of the canaliculus. Okay. And next, this is the intercellular surface which is facing the adjacent hepatocytes. Okay. Like this, we are having three surfaces of the hepatocytes. Okay. Next, there is a list of cells. Okay, list of cells. Till now, whatever we have come across, these are the list of cells which are present at the various part of the liver. Okay, already we have discussed each and every one, but to make it easy, I have added this chart. Okay, so I'll request everyone to go through this chart so that you will come to know what are the different types of cells, what is the structure, where they are present, and what is the function. Okay. Next, the most important part, that is the diagram, because in case of histology, diagram is the most important concern among the students. Okay, so this is the histology of liver schematic diagram. Okay, this is the schematic diagram of the liver under low magnification. Okay, so what you can see over here, first of all, you can see the cords of hepatocytes, right? See, these are the cords of hepatocytes. And you can see the central vein. In between the cords of hepatocytes, the spaces which are there, these are called as hepatic sinusoids. Hepatic sinusoids. In between the cords of the cells, the space which is there, these are called as hepatic sinusoids. Okay. Then some part of the septum can also be seen. Okay. Then at the angular interval, here you can see these are the portal triads. See, this is the portal triad. This is the portal triad. Okay. Now what is present inside the portal triad? For that you have to go for higher magnification. Okay. So under high magnification you can see the portal triad, the structures of the portal triad. Okay. So this is the schematic diagram of the histology of liver under low magnification. Next, here this is the histological image, okay, both under low and high magnification. That means when you will see the slide under microscope, okay, it will appear like this. Okay, so these things we will discuss in the histology class, in the practical class, don't worry. Next, this is the blood supply of the liver. That means not blood supply, the blood vessel which is supplying the liver. Blood supply of the liver means how the blood is flowing inside the liver. Okay, this blood supply of the liver means how the blood is flowing inside the liver. Okay, for example, if you remember the splenic circulation, do you remember the splenic circulation? Last time you have written in the exam also, splenic circulation. Okay, so in splenic circulation, what is the meaning? It is not which artery supply the spleen. This is not splenic circulation. Splenic circulation is inside the spleen, how the blood is flowing. That is the splenic circulation. So similarly over here, blood supply of the liver means how the blood is flowing inside the liver. Okay, so as you can see, portal vein and hepatic artery, we know two sources are there from which the blood is coming for the liver. Okay, so from the portal vein also, hepatic artery also. Then where they will go? They will go into the hepatic sinusoids, where the mixing of the blood is taking place. Next, where it will go? It will go to the central vein, then sublobular vein, then hepatic vein, and from the hepatic vein it will drain into the IVC, inferior vena cava, which goes into the systemic vein. Okay, this is how the blood is flowing inside the liver. Okay. Next, we'll discuss the microscopic structure of gallbladder quickly. Okay. So along with the liver, gallbladder is also there. Okay, and we have to study the microscopic structure of gallbladder also. Okay. Now here, as I said, you can't apply the concept of the layers of the GIT. Okay, it is not there. Now in case of a gallbladder, mainly three layers are present. What are those three layers? As you can see, mucosa is there, but here you will not see any submucosa. Okay, see, submucosa is not there. Instead of that, we are getting the fibromuscular coat followed by the serosa or adventitia. Now this part is similar, serosa or adventitia. Okay, serosa means the part which is covered by peritoneum, adventitia the part which is not covered by peritoneum. That part is same. Okay. Now there are some similarities, okay, in case of gallbladder. Can you see the lining epithelium of gallbladder? What is the lining epithelium of gallbladder? Simple columnar epithelium with striated border. That means microvilli are present. Okay. Along with that, lamina propria is there. Okay, in the lamina propria, connective tissue where blood vessels, nerves, are present, but the aggregation of lymphatic follicles are not present in the lamina propria. Okay. Next, fibromuscular coat is there. Okay, fibromuscular coat is there. Now this fibromuscular coat where we are getting the bundles of smooth muscle cells. Okay, and in between that we are getting the collagen fibers and elastin fibers. That is the fibromuscular coat. Okay, so submucosa is not there. Instead of submucosa, what we are getting? Fibromuscular coat. So fibromuscular means the smooth muscle cells are there. In between the smooth muscle cells, what is present? The collagen fibers and elastic fibers. Next, do you remember the arrangement of the smooth muscle fibers in case of GIT? How they are arranged in muscularis externa? Inner, circular and outer longitudinal layer. Okay, please revise. But in this case, no definite arrangements are there. That means irregular arrangements of the smooth muscle fibers are present in case of gallbladder. Okay, no regular arrangement, irregular arrangements are there. Okay, and in between that, what are present? Elastin fibers and collagen fibers, which together form the fibromuscular coat. Okay, and lastly, serosa or adventitia. Okay, so whatever we have discussed, it is mentioned over here. I will request everyone to go through it. You will understand. Let me show you the diagram. Okay, now here you can see. Here you can see the schematic diagram of the gallbladder. Okay, so this particular part is the lumen. Now don't misjudge this particular part with the villi. These are not villi. villi is absent, totally absent in case of gallbladder. Okay. Now here you can see this is the lining epithelium. So what is the lining epithelium? Simple columnar epithelium, that also microvilli are present. Okay. Next, this lamina propria, you can see this is the lamina propria. Okay, so this is the lamina propria, and then intestinal crypts are also there. Okay, crypts are also there. Lining epithelium dipping down into the lamina propria, crypts are also formed, and occasionally some glands may be present. Occasionally, not always. Okay, so this is the lamina propria. Now in lamina propria, blood vessels, nerves, these are present, but lymphatic tissue are absent. Next, you can see this is the fibromuscular cord. Now in the fibromuscular cord, you can see smooth muscle fibers are present. Okay. Now these smooth muscle fibers which are present, definite arrangements are not there. And in between that, you can see the elastin fibers, collagen fibers are present. And lastly, you can see this is the serosa. Okay. So this is the microscopic structure of gallbladder. Next, here you can see this is the this is under low magnification and this is under high magnification. Okay, of the histology of gallbladder. Okay. So that's all. Further under microscope we'll discuss in the histology class. Okay. Thank you.